Hello, you're welcome to this session at Medical Sciences Made Easy. And today we want to look at cell division, whereby we are going to introduce it and we look at the different phases of cell cycle. And this one is going to be covered under histopathology. We can see it in anatomy and physiology. So cell division, according to cell theory, we normally see that cells emanate or they develop from other existing cells. So we are going to see how cells, that even if I have one cell, this is one cell with a nucleus, how can I divide it to bring two? This is what we are calling cell division. So cell division is like multiplication or reproducing one cell duplicating into two cells. That is what we are calling cell division. So cell division is how cells produce other cells, or how, if I have one cell, how can I use it to produce two daughter cells, whereby one cell is able to produce two daughter cells, but all of them with identical, which are identical, having the same number of chromosomes as the parent. If this is the parent cell, we are seeing two daughter cells being produced. That is, and we are going to see different types of cell division, whereby we have said cell division is multiplication of cells or reproduction of cells, whereby cells multiply themselves to produce other cells which are either identical, or we can see under reproductive, whereby they have half number of chromosomes as the parent. And the types of cell division include we shall see somatic cell division, we shall also see reproductive cell division. So these are the major two types of cell division under cell cycle, which undergo cell cycle. And under somatic cell division, these somatic cells are found in all over the body except the germs, that is the sperms and the oocyte. So all other cells of the body except the germs are called somatic cells. So this somatic cell division occurs in the somatic cells of the body, whereby if I have a somatic cell, it undergoes what we call cell cycle. And at the end it produces two, whereby one parent cell produces two daughter cells, and these two daughter cells, they have the same number of chromosomes, and the same kind as the parent cell. That is in somatic. Whereas in reproductive, this one occurs in sperms and oocyte. So these are the germ cells. And these germ cells, for them, they undergo meiosis. And after undergoing meiosis, they produce daughter cells. And these daughter cells produced, they have half number of chromosome as the original cell. So if the original cell has 2n, we find the daughter cells, they have n. So meaning they are haploid. It produces haploid cells. Whereas under somatic cells, if I have parent cell which is 2n, we shall find also the two daughter cells is 2n. And meaning they are diploid. So this is the difference between somatic cell division and reproductive cell division. Please, as we continue, I always remind you to subscribe if you haven't subscribed, always like the video and share your comments. So as we are continuing with the types of cell division, we are seeing under somatic cells, and we know that in our body, the body of a human being contains 23 pairs of chromosomes. 23 pairs of chromosome, whereby out of 23 pairs of chromosome, 22 are autosomal. 22 chromosomes are autosomal, whereas the one pair is the one that is reproductive, is the one of the germs, which undergoes meiosis to produce daughter cells with half number of the chromosome as the original cell. So meaning 23 pairs, they give up what we call 46 chromosomes. So in our body we have 46 chromosomes, whereby the 45 chromosomes are autosomal, or they are somatic, they are found in the somatic cells, whereas the last pair, which is the 46, is found in the germ cells. And as we are continuing to see, we are going to see that somatic cells, for them, they undergo cell cycle, that is, they undergo cell cycle, and this cell cycle, it involves phases, whereby we shall see gap 1 phase, we shall see S phase, that is a synthetic phase, we shall see gap 2 phase, and lastly we shall see M phase. So we are saying that this somatic cell undergoes cell cycle, and this is the structure of the cell cycle, whereby the first phase is G1 phase or gap 1 phase. This one is a synthetic, or what we call S phase, and then this is G2 phase, which we call gap 2 phase. Then lastly is the M phase. So all somatic cells, all the 22 chromosomes, they undergo cell cycle, that is gap 1, S phase, gap 2 phase, and M phase. And if it is a 24-hour, if we divide it into 24 hours, we're going to see that gap phase takes between 8 to 10 hours, synthetic takes 8 hours, whereas gap 2 takes 4 to 6 hours. So M phase is the smallest. And if we can see everything that occurs in gap 1, why do we call it gap 1 or gap phases? We see in this phase there is no replication of the DNA. So the DNA remains the same number. And the cells in this phase, they are at rest. They are not replicating. So that's why we call it gap phase. That is gap. And cells undergoes this. And this cycle from gap 1 up to G2 is what we call interphase. So we shall see that interphase, part of interphase is the one that is covered by G1, S1, S phase, and G2. So this is what we call interphase. And when the cell goes into mitosis phase or meiosis phase, it starts from prophase. And we are going to see that in M phase, we are having interphase, we shall have prophase, we shall have anaphase, telophase, and sometimes cytokinesis. So we shall discuss in details the phases under mitosis, whereby we shall have prophase, anaphase, telophase, and cytokinesis. Whereby cytokinesis, cyto means cell and kinesis means movement, whereby the cell forms a furrow in the center to produce 2 identical daughter cells. So this is specifically the cell cycle. And if we can talk in details what happens in the G1 phase, in the gap 1 phase, we normally see majorly replication of the cytosolic components. We're going to see replication of cytosolic components of the cell, whereby these cytosolic components undergo replication, but not the DNA, only cytosolic components. So the requirements which the cell needs during gap 1 phase, or during this resting phase of which we are calling gap 1, there is replication of the cytoplasmic components. So we are seeing components within the cytosol, or what we call cytoplasm, undergoing replication, and we see even in the gap phase the replication of central, we see replication of centrosome. Replication of centrosome also begins at this phase. So in the gap 1 phase, we see majorly two things happening. Replication of the cytoplasmic content of the cell without the DNA replication or nucleus. Then the second one is replication of the centrosome, which begins in gap 1 phase. And there are cells which can fail to progress from G1 to S phase. And these cells, we call them, they undergo what we call G0. Undergo what we call G0 phase. And these cells that undergo G0 phase, or which take longer in gap 1 phase, they fail to replicate. And the cells which undergo G0 phase, cells take longer in gap 1, they undergo G0 phase, and those cells include the nerve cells. So meaning whenever a nerve cell is damaged, because the major importance of autosomal cell division, or what we call somatic cell division, is to replace the injured cells and to replace the dead cells. So whenever there is an injured cell, it is replaced by cell division, that is somatic cell division. And whenever there is a dead cell, it is also replaced by somatic cell division. So meaning the cells which undergo G0 phase, cells like nerve cells, once damaged, they are damaged permanently. So for them, when damaged, they undergo permanent damage, meaning they cannot be replaced or they cannot be renewed since they undergo G0 phase. So do not forget nerve cells, that for them, they undergo G0 phase. And that's why we are always careful when we are dealing with nerves, because once damaged, no repair, because they undergo G0 phase. So others progress into the S phase, or what we are calling synthetic phase. And in synthetic phase, we're going to look at it this side. Under synthetic phase, so under S phase, so this synthetic phase, for it there is only one major role being played under synthetic phase, which takes 8 hours. And in this synthetic phase, we are seeing majorly replication of the DNA. So we see there is majorly DNA replication in the nucleus, whereby DNA is undergoing replication. So the major role of the S phase is DNA replication, in that if I have this as my DNA, we are going to see this parent DNA undergoing replication to produce 2 daughter DNA. And these 2 daughter cells of the DNA produced during S phase, we shall find that they have one template strand of the parent DNA and another new synthesized strand. So these are the 2 daughter cells, or 2 daughter DNA formed, each with one strand, one template of the parent. And that is what we call semi-conservative theory under DNA replication. That this DNA replication, that there is unzipping or the opening of the strand, whereby each takes one strand, one from the parent and another one which is newly synthesized, which was defined by Crick and Watson under what we call semi-conservative theory. So in S phase, we see only replication of the parent DNA to give 2 daughter DNA, each with one template of the parent and another one which is newly synthesized. So that is the major action occurring under S phase. Then when cells finish S phase, they progress into the gap 2 phase. And this gap 2 phase, in the G2 phase, we are going to see majorly two things. One, we are going to see that more proteins and enzymes which are needed during mitosis are synthesized in the resting phase of GAP2. So under GAP2, we are seeing synthesis or replication of more enzymes and structural proteins required. And structural proteins. So we see it all happening in the cytosol or in the cytoplasm, whereby the cell during this resting period, if there is more enzymes needed during mitosis, there is more proteins or structural proteins and functional proteins required are synthesized in the GAP2 phase. And we saw in the G1 phase there was the replication of centrosome was beginning, and we see centrosome replication ending here. Centrosome replication ends, or it is completed in the G2 phase. So these are the major 2 points you can talk about under GAP2 phase of the cell cycle, which takes 4 to 6 hours when the cell is resting, forming more requirements which we shall need in the M phase. Then lastly, we can end with the M phase. And this M phase is the smallest, takes the smallest time because the cell take longer time in the interphase. We have said from G1 up to G2 is what we call interphase, whereby cell is preparing before from here. From here up to here is interphase, and is the one that takes longer time because cell is making sure all the requirements necessary are present before it begins to replicate under mitosis. So under M phase, we shall switch. You are going to see in the coming video that this M phase, for it takes short time, and it is divided into major 3 or 4 components. That is whereby it begins. The mnemonic I use is PMAT. PMAT. Whereby PMAT means we have prophase, we have metaphase, then we have anaphase and telophase. So whenever you want to remember this in their order, always remember PMAT. So PMAT, whereby it is prophase, metaphase, anaphase, and telophase, being the major component. So if we can be here, we can find this is prophase, this is metaphase, this is anaphase, then this one becomes telophase. Then later, the part of cytokinesis occurs in between anaphase and telophase. That's where we see cytokinesis, whereby it is just the making of the furrow or the depression in the center so that it divides into 2. So in between anaphase and telophase, we find also cytokinesis happening, whereby this cytokinesis, that if I have a cell like this at the center or at the metaphase plate, we see it making a furrow. It constricts at the center in preparation for making 2 identical daughter cells. That is what we call cytokinesis, which we shall see in details under mitosis. So as we conclude in this session of cell division, introduction to cell division and cell cycle, we have seen that there are major 2 types of cell division, that is somatic cell division and reproductive cell division. And under somatic, we have seen that cells undergo replication or they multiply to produce daughter cells which have identical number of chromosomes as the parent or as the original cell. And we have seen they occur in the somatic cells of the body except the germ cells. And then we have seen they undergo cell cycle, whereby they undergo what we call GAP1, S phase, gap2 phase, mitosis, and M phase. And this M phase contains that is PMAT, prophase, metaphase, anaphase, telophase, together with cytokinesis. And of course, this is what we must know about this. And under reproductive, we have seen this one occurring in the last pair of the chromosome, which is the germ. Find them in the germ cells, that is sperms in male and oocyte in ladies. And these guys, for them, they undergo meiosis to produce two daughter cells with half number of chromosome as the parent. So they have half number. Why do they undergo meiosis? Is that during fertilization, we are going to see one chromosome from the male and another one from the female joining together to form zygote. So we make them half, so that when the male produces the sperm and the ladies bring the oocyte, having half number of chromosomes, when they join together, they form a fully diploid cell. That is why the germ cells undergo meiosis, not mitosis. So under mitosis, under M phase, if it is somatic cells, they undergo mitosis only. And if it is reproductive cells, for them, they undergo both mitosis and meiosis to produce half number of chromosome as the original parent. Thank you so much for being with us. Always remember to subscribe. Always put your comments for more learning to happen. Thank you so much.